Yeah. So coming to this chapter, so we know that the discovery of the cell was very important landmarks, especially that happened with the discovery of microscopy. So we'll discuss about microscopy and the evolution of microscopy in the later sessions when we are very close to the examination. But coming to the core of the concepts, so once the cell was discovered by the Robert Hooke, and then it was given names. So we know that gradually what happened is we came to an understanding that all living organisms are made up of cells and product of the cells. And in generally, when we talk about the function of the cell, we know that all living organisms from the bacteria to the complex eukaryotes, we have the same biomechanical pathways, like the functions of producing proteins, expression of genetic material, transportation of substances, maintaining cellular environment. All of them happens evenly across all genera of the kingdoms. So that's what gives the cell a title of cell being a fundamental and structural functional unit of all living organisms. So with this, the great curiosity was originated, as we know, in the late 19th century, where people were very curious about what lies under the microscope. So as all the scientists and all these botanists, zoologists were racing to see what is cell and understand what is cell, one of the greatest contributions come from Sheldon and Schwann, where they started proposing something called as cell theory. So we know that the Sheldon is a botanist and Schwann is a zoologist. So Sheldon, he observed all the plant components and the plant cells, and he understood that the plants have a very rigid cell wall, and within the rigid cell wall, they have this membrane, okay, which protects the internal environment of the cell. Okay, so once we have the Sheldon's observation of the plants, and then comes Schwann. Schwann actually observed both plants and animals. He was a zoologist basically, but he ended up observing both plants and animals. Now Sheldon and Schwann comes together at the table and then they discuss their observations and eventually they come up with the theory which we call it as a cell theory. Now when we talk about the Sheldon and Schwann's proposal of the cell theory, they just told that all living organisms are made up of the cells and the product of the cells. So this is the first statement of our cell theory. Now, they failed to explain how the cells give, how the new cells come up or how the cells were born. So that was clarified by Rudolf Virchow. So, omnis cellulae e cellula, that's what the statement that changes. So he says that the cells arise from the pre-existing cells. To complete this theory, that is a cell theory. So cell theory goes up with these two statements. One is all the living organisms are made up of cells and the product of the cells. The cells arise from the pre-existing cells. Correct. So this is the modern cell theory that they proposed. Now, when we look at the cells can be divided into two major categories. One is a prokaryotic cell, other one is a eukaryotic cell. Now, the reason why we have classified this prokaryotic and eukaryotic cell is because of the complexity of the cell itself. Now, when we look at the prokaryotic are called as primitive cells or the early cells. Okay, screen is not fully seen. Hold on. Is it? So then give me a second, students. Okay. Maybe is this okay? Just give 1 second. Now also it's not seen, right? Yeah. I hope now it's visible. Okay, let me look at it. Yeah. Yes. Perfect. Thank you. Yeah. Yeah. Okay. Okay. So in Krishna's account, two people are present. Ashta and Krishna both are present. Okay, I know that, guys. Thank you. Okay. Chalo. So when we look at the prokaryotic cells, the complexity is quite poor, and the pro starts with early or old. We can call it as early or we can call it as old. So carry-on translates to nucleus. They have a very early nucleus or they have a very old nucleus compared to that of the advanced nucleus. So the prokaryotes, one of the most common factors of the characteristics of the prokaryotes is that all prokaryotes have the cell wall surrounding the cell. Now, this cell wall characteristics is very unique to the prokaryotes. Why? Because the composition of cell wall itself is quite different. Now, when we look at what is within the cell wall contains a plasma membrane. Within the plasma membrane is the protoplasm or the cytoplasmic mass. Now, here, the cytoplasm is quite similar to that of, okay, the cytoplasm is very similar to that of your eukaryotes. If you look at the cytoplasm of the prokaryote, what we can understand is the prokaryotes lack any membrane bound organelles. If I say any membrane bound organelles, that means one of the major membrane bound organelles that we come across is mitochondria and chloroplast. Correct, right? So they don't have this mitochondria as well as the chloroplast. So whereas whatever the other membrane bound organelle, that is the nucleus, even the nucleus is quite absent. There is no well-defined nucleus. Why do we call it as absence of well-defined nucleus? Because the nucleus do not have any nuclear membrane. And the nucleus and the components of the nucleus are just floating naked in the cytoplasmic mass of the prokaryotic cell. Now I have ribosomes, and the ribosomes are of specific type, that is the 70S type of the ribosome. Correct, right? So they have 70S type of ribosomes. I'll mention it here. And they don't have, as I mentioned, they don't have mitochondria, they don't have chloroplast or any of the plastids. Correct? So eventually, when you look at a prokaryotic cell or a characteristic of a prokaryotic cell, one thing that we have to closely observe is the genetic material. So the genetic material is basically circular and it is double-stranded, and they are closed and naked. So that is why they are called as nucleolus. Okay, nucleosol. So now the prokaryotic cell also houses an extracellular, that is extra chromosomal genetic material, which we call it as a plasmid. So the properties of the plasmid and the role of the plasmid we have studied in biotechnology. Plasmid is nothing but an extra chromosomal DNA which is outside the nucleus. That is why we call it as extra chromosomal. Why it is important? Because we know that the plasmid gives rise to the characteristics of the bacteria, such as antibiotic resistance character. So that is why we consider the plasmid DNA, right? So there are double-stranded circular DNA which is outside the chromosome. And then, as I was talking about the envelope, the cell envelope, very important topic. When we talk about the cell envelope of the prokaryotes, they have three layers. The outermost layer is a capsule, okay, and which is called as a glycocalyx layer. Below the glycocalyx layer is the cell wall, and within the cell wall is the cell membrane. Now, this glycocalyx layer, the property of a glycocalyx layer is very, very important. See, either they are thick and tough layer or they are slimy layer. So why glycocalyx is very important? Because this glycocalyx layer is what gives the protection for the bacteria against the antibiotics. That's very, very important. Okay. Now you might have heard about this rough strain and the smooth strain characteristics of your, Streptococcus pneumonia. So the rough strain and the smooth strain comes because of the glycocalyx or the capsule properties. The smooth strain have very smooth and slimy glycocalyx layer, whereas the rough strain don't have any glycocalyx layer that makes its cell wall looks very rough. Now coming to the cell wall, the cell wall of the bacteria is very, very important. Why? Because it's a single cell that performs all the living activities as us. So that is why the internal cell environment is very, very important. And the cell wall gives the mechanical support for our bacterial cell without collapsing on itself. And now the cell wall is a very important microbiological property for us to understand the categories of our bacteria by doing one important staining technique called as Gram staining. So the Gram staining is performed on the bacteria, and we divide the bacteria based on the results as Gram positive and Gram negative bacteria. Now, why this is very, very important? Because the cell wall of a bacteria is not made up of cellulose. It is made up of a substance called as peptidoglycan. Okay. So this peptidoglycan is also called as murin layer. See, so the peptidoglycan is an overall composition, but as the murin gives the mechanical strength to the cell wall. Now, when we perform the gram staining, now based on the gram staining is taken up or not taken up. If the cell has taken up, it appears violet or blue, okay, something bluish. Whereas if it does not take up the gram stain, the cells will start appearing pink. Now, this is a very important categorization of bacteria because we come to know with the interaction of bacteria that mostly gram-negative bacteria are the ones that causes most of the diseases. Okay, so coming to other important characteristics of the bacteria, if you look at the plasma membrane of the plasma membrane of the bacteria is very similar to that of the eukaryote. So we'll discuss it later. If you see the inner foldings of the plasma membrane, see there are lots of folds, okay, within the plasma membrane of the bacteria that gives rise to a higher surface area. So it increases the surface area for a reason. So that inner foldings of the plasma membrane is called as mesosomes. Remember this name. A lot of questions have been asked. Prokaryotes lack mitochondria, but which cellular organelle or which cell part of the bacteria performs the activity of mitochondria? So mesosomes act like the mitochondria of prokaryotes, where the cellular respiration will take place, and mesosomes help in the formation of ATP. And other functions of mesosomes is they help in cell wall formation, they help in DNA replication. Okay, respiration is where you produce your ATP. They help in secretion process like cell vomiting and cell engulfing. Okay, and they also increase the surface area of plasma membrane, and they help in increasing the enzymatic content. More importantly, you have to remember that mesosomes perform cellular respiration. Okay, so one of the important functions of mesosomes. And another important characteristics is presence of chromatophores. That is, when you look at the blue-green algae, okay, so they have photosynthetic pigments, okay. Example, xanthophyll, and then you have chlorophyll A, chlorophyll B, and then you have monoxanthin. So these are some of the pigments that are present within the chromatophores of your cyanobacteria or the bacteria. Now, if you look at the bacteria's hold on, if you have it, okay, the bacteria's entire morphology can be divided. Okay, I'm telling bacteria because all the bacteria are prokaryotes, and prokaryotes include only monera. So we know that the bacteria is classified into four different types based on its shape. One is cocci, bacilli, spirillum, and vibrio. So this will give the morphological basis of classifying the bacteria. For example, we have Streptococcus pneumonia. Streptococci talks about how the bacteria is arranged. For example, we have Staphylococcus pneumonia. So if I talk about Staphylococcus, that means they are arranged in groups of 3, 4, etc. Are you understanding? So monococcus means if you look at the cells of cocci, they are arranged as single cells. You understand? Based on how the colony is there, based on the shape of cells, we can categorize the bacterial cells. So we have cocci, spirilla, vibrio, and then we have bacilli. So these are the morphological categorization of the bacteria. If you look at the external surface of the bacteria, you can see that the bacteria also have cell wall extensions like flagella and cilia and pili. Okay, so they also have a very important role to play. If you look at the bacterial flagellum can vary from 1 to 2, and it can, for example, if you look at the bacilli, it can be like this, it can be like this, it can be on the both the sides, or a bunch of flagella on one side, etc. Okay, so though that can also vary. So based on number of flagella present, based on the position of the flagella, we can categorize the flagella. What is flagella? Flagella is the cell wall extension of the bacteria that helps in motility. A flagella can be divided into three regions. One is basal body, hook, and the filament. The basal body is within the cell wall, and the hook is an extension of the cell wall, and the filament is a long whip-like structure where the spinning of the filament gives rise to the motion of the flagella and with return to the motility of the bacteria. Okay. Now we'll discuss about the structure of the flagella. So if I take a cross section or electron microscopy image, what I can understand is I have divided the flagella into three regions, that is basal body, hook, and the filament. If I look at the basal body of the bacteria, they are made up of very important protein motors, okay, which are called as L rings and S rings. So these rings, okay, We'll talk about it even more detail, but for now, these rings enclose this rod, okay, a protein rod. So these rings and the rod, they act like a protein motor, okay. One part of the ring is in the plasma membrane and another part of the ring is in the cell wall. So they are within the cell wall itself. Arising from this, they give an extension called as a hook, and hook is a bent short anchor that attaches the basal body to the filament, okay. Now along with the hook, the filament, along with the hook and the filament, the entire structure will rotate. That will give rise to the motility of the bacteria or movement in the bacteria. Now, another things I told you, that is there are pili and there is cilia, etc. Now, if you look at the cilia and fimbriae, okay, which are another two structures. So pili is something called as a small cell wall extension, okay. So pili is also called as sex pili. So you might have heard about a process called as conjugation, where the two strains of bacteria, they come together and they build a bridge, and the DNA material is exchanged between these two. How does this happen? This pili will attach to the other bacteria and the genetic information is transferred between these two. So that is a pili help in a type of sexual reproduction called as conjugation. Got it? Whereas fimbriae is a brush-like structure, okay, which is again the cell wall extension. Now what this fimbriae will do, usually in the flowing streams or running water, what happens is these bacteria have to have an anchorage, or they have to attach to a substrate. For example, let's say you have any of the disease causing bacteria, and then they have to live within the host, okay. So if these parasites have to live in the host, they have to find a way to attach to the host tissues. So they anchor to the host tissues using this brush-like structure called as fimbriae. Understood? So eventually what will happen, you have some of the extensions such as flagella, pili, fimbriae. Now coming to cilia, and oh sorry people, I forgot that pili is made up of special protein called as pillin. Okay, pillin proteins. This is very important. Please note it down. Okay. Now coming to cilia, cilia is a bristle-like structure, like your toothbrush. So there might be thousands of cilia across the cell wall of a bacteria. Now they are very mechanically oriented. That means you can see that the boat race, all of them are rowing together. Similarly, the cilia also rows together. That means the rowing movement is very synchronized with other cilia, and that will help in the movement of the bacteria. Whereas cilia and the flagella structure are very, very structurally similar. Okay. Now coming to, as I told you, ribosomes are 70S. I'll talk about the properties of the ribosomes. Just give it a second. Okay. So whereas there are inclusion bodies in the prokaryotes, very important thing, and they are very freely lying in the cytoplasm. They do not have any membrane bond organelles. Example, phosphate granules, cyanophycean granules, glycogen granules, and gas vacuoles are some of the examples. Very, very, very important point, students. Okay. So are inclusion bodies present in our bacteria? Answer is yes. What kind of inclusion bodies are they? They are non-membrane bond organelles, and they store some of the substances like phosphate, cyanophycean granules, glycogen granules, gas vacuoles, etc. Very important. I know that it will mess up with your brain. I know that you will get confused. Okay, is it in eukaryote or prokaryote? But remember, inclusion bodies are in prokaryotic cells. Clear? Now, coming to the eukaryotic cells, the modern cell, understanding an eukaryotic cell will give us a very complete detail about how the cell functions, okay, or what is the cellular properties that exist. So let's take a look at it. Now, the cell membrane is a beautiful structure. Why? Because the property of the cell membrane is what enables the cell to maintain its internal environment to that of the external environment. Correct, right? So now we know that the composition of the cell membrane is a very key aspect of how the cell membrane functions. Now, if I ask a cell membrane, you will immediately tell that the cell membrane is nothing but a semi-permeable membrane. Correct, right? And it semi-permeability means what? It allows only that is necessary, and it does not allow something that is not required. As simple as that. Now, how does this do that? Now, if I look at the composition of the cell membrane, we call that semi-permeable membrane or a plasma membrane as a lipid bilayer membrane. That means the major composition should be lipid, but you're missing something. Along with the lipid, proteins are also a very, very important component of our plasma membrane. Got it? So we have lipids and we have proteins. Do we have any other thing? Yes, there might be fats, there can be cholesterol, okay, etc. Now coming to the function of the, like the properties of the cell membrane. The cell membrane, it is made up of lipid bilayer, I told you. Now, if I take a normal RBC cells, now you can see that 52% of the RBC cell is made up of protein, 40% is made up of lipids. This ratio can change. The lipids can be 70% to 20%, vice versa, that can happen to the protein also. It completely depends upon the function and the role of the cell, okay, the nature of the plasma membrane. Now, as I was telling you, if I take an RBC cell, very important, remember the composition of the RBC cell is made up of what and what. So they will give dash amount of protein and dash amount of lipids. What is your answer? Should be 52% of protein and 40% of lipids. Yeah. Now, if I say that it is 52% of protein and 40% of lipids, let's see how they are arranged. So we know that the semi-permeable model, or it is also called as, the plasma fluid mosaic model of the plasma membrane, will give us an idea of how the arrangement of plasma membrane is done. Now, if I see the type of lipids that are present in the plasma membrane, they are made up of phospholipids. Correct, right? Now, what is the property of a phospholipid? A phospholipid is amphipathic in nature. What is amphipathic, people? If I write amphipathic, what is amphipathic? Means it acts like an amphibian, like a frog. Okay, I'll give an example as frog. What does frog do? It lives on both land and water. Similarly, the lipid molecule also act like a frog. That means it allows uh, both natures. Like I love water and I hate water. That means it is both hydrophobic and hydrophilic in nature. So if it is both hydrophilic and hydrophobic in nature within the same structure, we call it as amphipathic molecule. Now, what is this amphipathic molecule? We know that the phospholipids have two important ends. One is a phosphate end, other one is a fatty end. For example, if I write a structure of a phospholipids, we know that it is made up of glycerol, three carbon atoms. Okay, to that a phosphate molecule is attached, and to the carbon, two chains of fatty acids are attached, and unsaturated fatty acids are attached. Now you can see that there is a phosphate head. Okay, and then you have a fatty tail. Correct? No, people. Now we know that this fatty acid tail is hydrophobic. Okay, it is hydrophobic. Whereas if you look at the head of the phospholipid, it is hydrophilic. Very beautiful arrangement. Because of this molecule is where the property of the plasma membrane depends majorly. Now you can see that all the hydrophilic, that is the heads of your phospholipids, are pointed to the outside of the cell and towards the inside of the cell. What is the reason? Because the external environment is highly prone to the interaction of water. Your body is made up of 70% of water. The environment is made up of 70% of water. So water can degenerate or it can disturb the cellular environment. So it has to protect the inside of the cell. Now, if I look at the insides of the cell, the internal environment of the cell is also made up of majority of water. So it wants somebody to interact happily with the water content that is in the cell and not disturb the movement of particles across the cell. I understand, right? So outside also there is water and inside also there is water. So we want something that interacts with water and do not disturb the cell environment. So that is why all the water interacting molecules, that is our hydrophilic heads or the phosphate heads, are pointed towards the outside and the inside of the cell, whereas the tails are projected towards the inside. You can see all the tails are projected towards the inside. Now, if I look at the tails of this fatty acid chain, they have this positive and negative interactions that is happening. Correct, right? So they have this carboxyl ends and they have this positive ends, okay? The positive and negative ends. So these tails, and these tails, they have opposite charges. So these opposite charges will attract these tails and they hold in position. We know this type of ionic interactions are not like covalent bonds. They don't bond tightly. That is what gives these interactions to make these two layers go slide upon each other. Understood? They can move upon each other. They can just slide upon each other. So this is the way in which it just maintains the environment of the cell. Another important aspect of the plasma membrane is the movement of substances. Plasma membrane requires to maintain environment. For maintenance of environment, sometimes it requires to throw out the some things that is not necessary to it, or transport something which is very, very valuable, or allow some things which such as raw materials or important food materials on the outside. So all this has to happen. So that means transportation has to happen on a very regular basis. How does it support this process? That support comes from the protein integration. So I told you that it is made up of two major components. One is the protein component, other one is the lipid component. We discussed about lipid. Now the protein, how does it play? Now, if I look at the protein integrates within the plasma membrane in various ways. Some of them are very superficial. When I say superficial, they are just attached to the surface of the protein. We call it as, okay, do we have this? Yeah. We call it as peripheral proteins. Yeah, here it is, sorry. So you can see that it is peripheral protein. What do peripheral proteins do? They're just barely embedded to the structure of the plasma membrane. And some other proteins, they are integral proteins. What do? They completely puncture both the lipid layers and they create this channel. And these channels are very heavily guarded. Okay. So some of them are very peripheral, some of them are channel proteins. That means integrated proteins. Integral proteins, we call it as. So these peripheral proteins, what do? They peripheral proteins, the role is to act like receptors. For example, if you take any medicine, if you have to stimulate a cell, if you have to receive a signal or anything, these peripheral proteins, they play a very important role. Whereas what happens to these channel proteins? So these channel proteins or carrier proteins, for example, I want to push sodium and potassium. In order to push sodium from inside of the cell to the outside of the cell, I do not allow something very easily. So what happens? I will make sure that you go through the gate. So these channel gates or carrier proteins, what they do is, okay, you want to go outside, give me some pass or money. So now we have to spend some ATP. The ATP will make a configuration change, opening this channel, allowing this sodium to go out. Understood? So what kind of molecules can easily interact with the plasma membrane and what kind of molecules cannot easily interact with the plasma membrane? Very, very simple question. And this question is very valuable question as well. So for that, answer is very simple. If I have a hydrophilic molecule, if I have a water interacting molecule, these molecules will not easily interact with the plasma membrane. Why? I can cross the hydrophilic layer, but between there is a hydrophobic layer. So when I have a hydrophobic layer, obviously what should I do? I should go and ask the channel proteins or the carrier proteins, see, I have to be transported within. Please help me. Understood? That means, is it so that all the water molecules that crosses this plasma membrane have to pay money or have to go through these gates? Definitely no. The movement of water majority happens because of osmosis, which is a passive transport. Okay, there are many ways of transport. We have diffusion, we have facilitated diffusion, we have osmosis, and we have active transport. So different means of transport happens facilitating individual substance. For example, we have glucose. Okay, glucose is very important for us, right? Very, very important for cellular energy. How do you think the glucose is transported? Glucose is very smart. I am energy. If I spend energy to go inside and give energy, that means I'm wasting a lot of energy. Correct, right? So that is what glucose does. Glucose takes the help of sodium. So glucose follows sodium. So you go and try to attach to the sodium gate or a sodium channel protein. So what sodium will do? Sodium will spend an ATP. When it spends an ATP, the channel will open. Now what will glucose do? Glucose just follows sodium and then gets an entry into the cell. This is called as facilitated diffusion. Are you understanding my point? Right? So like this, many substances basically are transported with several means based on its property. What is a simple property? If you are hydrophilic, you have to go through the gate. If you are hydrophobic, you can easily interact. So what are the hydrophobic molecules that easily interact? Take for example your hormones. Okay, sterols, for example estrogen, testosterone, or you have your cholesterol, lipids. they can easily interact with your lipid bilayer and they can make an entry. Understood? So that is why it's very, very important to understand property of plasma membrane. Are you following, students? Yeah. Are you understanding? Guys, am I audible? Yes, no? So next one is the cell wall. So we know that the cell wall is a non-living material that is present in the plant cells, fungi, okay, and then protists. And then we don't have, in protozoans, we don't have cell walls. Whereas in prokaryotes, we have cell wall, and we discussed that the nature of the cell wall varies, correct, right? So when you talk about the cell wall, it is non-living. It is basically made up of cellulose and hemicellulose and some of the minerals like magnesium, calcium, etc. So this cell wall basically, they prevent damage, they prevent infections, they prevent any undesirable material and gives mechanical support to the to the cells, etc. So usually the composition of cells has cellulose, hemicellulose, pectins, and then it has proteins. Okay. So coming to the eukaryotic cell wall, the eukaryotic cell wall has two different stages. One, it can appear as a primary cell wall, usually in the young cells which are just born or it's just very young. Okay, and it is still in the dividing stage. You can see the primary cell wall. What is the nature of primary cell wall? It is flexible. It can change its shape. It can be moved. It can be divided, etc. So how does this property change? Because primary cell wall is made up of more. Sorry, I'll write it. It is made up of more hemicellulose. Hemi hemicellulose and less cellulose. That is what gives the flexible nature for our primary cell wall. Later, what happens is from the inside, see from the inner side, this primary cell wall, see this is a plasma membrane. From the inner side, what happens? A new cell wall starts building up during the process of maturation. So when the cells stop dividing, the primary cell wall will be replaced into a secondary cell wall where it is reversed. There is high amount of cellulose and less amount of hemicellulose. Understood? There is less hemicellulose. So this will make the cell wall very, very rigid. Now the secondary cell wall is very, very hard, and these cells do not divide, and they are mostly found in mature non-dividing cells. Now, if I take one cell and if I take another cell, these cells are cemented between each other. That means there is an attachment between the cells. And that attachment comes through something called as middle lamellae. Very important component that cements two cells or two cell walls together. So this middle lamellae is composed of calcium pectate, okay, and it holds all the neighboring cells together. Okay, so there is something called as plasmodesmata bridges. You might have heard about it. Usually, you can find it in fungal cells and even in the higher plants. What happens is the cell wall and the plasma, the cell wall and the cytoplasm of the two cells are interconnected through some bridges, and these bridges are called as plasmodesmata bridges or cytoplasmic bridges. So basically, they exchange material or exchange genetic information, etc. Okay. And the next one we have to talk about is the endoplasmic reticulum. Very important organelle. So endoplasmic reticulum, Golgi apparatus, vacuoles, lysosomes, they all come under a systematic unit. We call it as endomembrane system. So this endomembrane system, why do we call it as? Because each of the components, they contribute to something. They're all interdependent. So whenever you see an interdependent system, we call it as an endomembrane system, and that appears in our cell. So endomembrane system includes what? Golgi apparatus, endoplasmic reticulum, lysosomes, vacuoles. Okay, so these are the components. So let us talk about endoplasmic reticulum. Endoplasmic reticulum, if you look at it, they are very close to the nuclear membrane. First thing, you can see that this is the nuclear envelope, and you can see that they are quite scattered and they are very large in number across your nuclear region. So that is their speciality. Now, if you look at the endoplasmic reticulum, what you can see is there are three important components. One is called as cisternae, cisternae, tubules, and vesicles. So they appear in three forms, okay? Majorly they appear as cisternae and tubules, not vesicles. So this, what is cisternae and what are tubules? If you see any of the flat disc shaped structures, they are called as cisternae. Understood? What are tubules? You can see here, each of these narrow tubes are interconnected into network of tubes. So these are called as tubules. So they're made up of two important components. One is cisternae, other one is tubules. Okay. The role of endoplasmic reticulum, what it does is it has an association with ribosomes. If the endoplasmic reticulum is associated with the ribosome, they start appearing rough, and we call them as rough endoplasmic reticulum. And some of the endoplasmic reticulum, they are devoid of any of the ribosomes. If they don't have ribosomes, they are called as smooth endoplasmic reticulum. Now, what is the function? Based on the association of ribosomes and not, they can be classified based on the function. For example, if you talk about smooth endoplasmic reticulum, they help in lipid synthesis, steroidal hormone synthesis, etc. Whereas if you talk about the rough endoplasmic reticulum, since ribosomes are associated with them, they majorly participate in protein synthesis. Okay. Now, hold on. And the next one is Golgi apparatus. What's the time? Chalo. We'll just take a pause here and then let us have some interaction. Yeah. Okay, here's your first question, students. Okay. Which of the following is carried out by the cytoskeleton in the cell? Transportation, nuclear division, protein synthesis, and motility. Let's go. Let's answer this question. What is the function of cytoskeleton in the cell? What is the function of cytoskeleton in the cell? Is it transportation? Is it nuclear division? Is it protein synthesis? Or is it motility? A, B, C, D. Guys, what's your answer? Make it fast. Motility. Exactly. So what are cytoskeleton? Cytoskeleton are made up of microtubules, right? So these microtubules, they are arranged within the cytoplasmic mass and they are oriented in different fashion. If the cell requires motility, for example, if it wants to change its shape or if it wants to squeeze and then move towards the tissues, for example, you have phagocytes. So their cytoskeleton, what they do is they help in change the shape of the cell and then they eventually help in motility. Transportation, protein synthesis, and nuclear division is not the role of it. Okay. Chalo. And the next question is, which of the following are not considered as a part of endomembrane system? You have mitochondria, endoplasmic reticulum, chloroplast, okay, Golgi apparatus, and peroxisomes. So, okay, let's see. So we have A, D, and E only, B and D only, A, C, and E only, and then you have A and D only. Which of the following is not considered as a part of endomembrane system? What's your answer? I always tell my students whenever you get questions like this, okay, try to answer within 10 seconds. Straight and easy. Correct? Okay, you have answered. C. He says A, C, and E only. How many agree? So he says mitochondria, and then he says C is chloroplast, and E, peroxisomes, are not, you see, here is where people get confused. Not considered as a part. And he's absolutely right. Mitochondria is a semi-autonomous organelles. Chloroplast is a semi-autonomous. That means they can happily function on their own even without the cell. Okay, so chloroplast, they're semi-autonomous, and peroxisomes, they are not a part of your endomembrane system. So endoplasmic reticulum and Golgi apparatus are the part of your endomembrane system. Very good, students. Okay, let's go make it faster. Given below two statements, in bacteria, the mesosomes are formed by the extensions of plasma membrane. The mesosomes in bacteria help in DNA replication and cell wall formation. In the light of the above statements, choose the most appropriate answer for the given options. Statement 1 is correct, but the statement 2 is incorrect. Statement 1 is incorrect, but the statement 2 is correct. It should be 2. Okay, I'll just make change. Both statement 1 and statement 2 are correct. Okay, and then you have both statement 1 and statement 2 are incorrect. What's your answer? Already we have. So 42. That means now you have answered, Jaideep. There are two options, B and C. What should I consider? Is it B or C? Is it B or C? It's for the second one. That means, okay, not for it. Now you're making me confused. Okay, what's the answer for this now? Mesosomes are formed by the extensions of plasma membrane. Mesosomes in bacteria help in DNA replication and cell wall formation. Isn't it right? Yes. I will answer this question. Your time is up. Yeah. So see the first question. Mesosomes are formed by the cytoplasmic extensions. We just read about it, right? They help in cellular respiration. They help in cell wall formation. They help in DNA replication. They help in increasing the enzymatic activity. They help in increasing the surface area. Very, very important question. It's a 2023 question. Okay, both statement 1 and statement 2 are correct. So that is option C. Okay, let's go. So now we have two columns. We have to match the column 1 with column 2. Okay, what is metacentric chromosome? We have metacentric, acrocentric, submetacentric, and telocentric chromosomes. And then we have the explanation for it. So chromosomes situated close to the end forming one extremely short and one very long arms. Centromeres at the terminal end. Centromere in the middle forming two equal arms of chromosomes. Centromere slightly away from the middle forming one short arm and one longer arm. Okay, now let's go one by one. I know that match the following takes a little bit of time, and I know that you can get the right answer as well. It's easy question. When I talk about metacentric, that means the centromere is in the center and it divides the chromosomal arms into two equal halves. Right? They are called as metacentric chromosomes. So I'll write it as A here. Acrocentric. Okay. See, some people are very oriented. Some people are can remember anyways. Let's go for submetacentric because that's the order we studied. Metacentric, submetacentric. Now, what happens in submetacentric? Two longer arms and two shorter arms. That means the centromere is slightly away from the chromosome. Correct, right? So, what do we have? Centromere is situated close to the end, forming one extremely short. No, this is not the one. Chromosome is slightly away from the middle, forming one short arm and one longer arm. That is called as submetacentric. So, I'll put it as C. What about acrocentric? Acrocentric is this, where you have the centromere very close to one end and forming one very long arm and very short arms. So that is here. Correct, right? Am I right? Somewhere I made a mistake. Okay, B. This should be B. Okay, next one. Telocentric is what? Telocentric is you have your centromere at a very end, leaving to one extremely long arms. But if you see the centromere, they're at the one terminal end. So that will give us D. Clear? Yes? Yeah. Can we go for the next one? So we have one more. This you have to answer, students. In column 1, we have chromoplast, amyloplast, aleoplast, and alluroplast. And in column 2, we have proteins, oils and fats, starch, and keratin. Okay. Now I will tell one option you should tell. What about chromoplast? Is it 1, 2, 3, or 4? Chromoplast. What do they store? Guys, you can see the question, right? Very clearly. Can you see the question? Try to answer this. Nizamabad students, Jaideep, Vijayendra. Come on, guys. What's the answer for this? What do you study about this? Yeah, it's a pigment. So it's a pigment means can I go for chromatophores as chrome bearing pigments or chrome storing pigments or bodies? So I'll write it as keratin. Keratin is a pigment. So A. What about amyloplast? What about amyloplast? Amylo means what? What about amylo? You just have to mention one, two, three, or one, two, three. Just mention one, two, three. So amyloplast, three. So he says amyloplast stores starch. Absolutely right. How do we remember this? Amylo, salivary amylase, right? Salivary amylase. So when we talk about salivary amylase, you will get what? Starch. Okay. So they store starch. Next one. What about elioplasts? Elioplast. One. Elioplast are protein, absolutely right. And that is left with alluroplast, which is oils and fats. So what are we discussing about, kids, here? We are talking about plastids, okay? And we have three types of plastids. What are those? One is chloroplast, chromoplast, and leucoplast. So we know chloroplast is a photosynthetic body, and chromoplast is a pigment bearing. For example, keratin is a pigment. And coming to leucoplast. In leucoplast, we come across three types of leucoplast. One is amyloplast, alioplast, and alluroplast. And here your answer. Amylo for starch, elio for proteins, alluro for oils and fat. Clear? Good. Thank you for the response. Let's go for the next question. Given below are two statements, and we have the first statement, that is mycoplasma can pass through, less than 1 micron filter size. Statement 2 is mycoplasma are bacteria with cell walls. In the light of above statements, choose the most appropriate answer for the given below. So the same thing, statement 1 is incorrect, but statement 2 is correct. Statement 1 and statement 2 are correct. Then you have both statement 1 and 2 are incorrect. And then we have statement 1 is correct, but statement 2 is incorrect. So it's based on mycoplasma, PPLO, pleuropneumonia like, yes, organisms. So is it right that the mycoplasma can pass through a filter paper that is 1 micron less than 1 micron? Yes. So the statement 1 is correct. What about the statement 2? That is mycoplasma are bacteria with cell walls? No. That means what? They don't have cell wall, right? That is why it is very hard for us to create antibiotics for this particular organism, and it's very, very dangerous. Okay. So that gives us what? Statement 1 is correct and statement 2 is incorrect. Correct. So that is our option D is the right answer. Okay. Next one. Which of the following statements with respect to endoplasmic reticulum is incorrect? SER, smooth endoplasmic reticulum, are the sites for lipid synthesis. Then you have rough endoplasmic reticulum has ribosomes attached to the ER. And then smooth endoplasmic reticulum is devoid of ribosomes. And finally, we have in prokaryotes, only rough endoplasmic reticulum are present. What do you think? Which of the following statements with respect to endoplasmic reticulum is incorrect? What's your answer, guys? Very easy one. I'm waiting. So, can we answer? Yep. So it's very easy. So smooth endoplasmic reticulums are the site for lipid synthesis. Yes, this is true. That is what they do. They synthesize lipids as well as sterol hormones. Rough endoplasmic reticulum has ribosomes attached to it. Yes, that is what makes them look rough. And they're the site for protein synthesis. Smooth endoplasmic reticulum is devoid of ribosomes. That is also true. In prokaryotes, only rough endoplasmic reticulum are present. No, both rough endoplasmic as well as smooth endoplasmic reticulum are present, and you can see that the endoplasmic reticulum are freely floating in the cytoplasm of a prokaryotic cell. Don't confuse that. Okay, prokaryotes don't have ribosomes. Oh, sorry, endoplasmic reticulum they do have. Okay, chalo. Next one. Statement one. Membrane bound organelles. This is a statement one. Okay, membrane bound organelles of the endomembrane system coordinate cellular functions. Mitochondria and chloroplast are not considered as a part of endomembrane system. I think this question is quite easy, so let's skip it. We discussed about it. Okay, here it is. Which type of substances would face difficulty in passing through the cell membrane? One, substances with hydrophobic moiety, substances with hydrophilic moiety, substances irrespective of hydrophilic and hydrophobic moiety, substances which are soluble in lipids. So the question is, which of the substances would face difficulty in passing through the cell membrane? Didn't I tell you? This is 2023 question, okay? When I picked this question, uh, I've seen this many times coming on the nature of substances that cross the plasma membrane. So what's the answer, guys? We just discussed this. It should be easy. Quick answer. Come on. Guys, make it fast. No answer. I wish we had polls. Anyways, so okay, which of the substances will face the difficulty? As we have already discussed, it is always hydrophilic molecules. So whenever there is hydrophilic molecules, they find it very hard to cross this hydrophobic tails, right? So here, though, there is hydrophilic heads, that is the phosphate heads. They will interact very easily here, but to cross the hydrophobic ends, it's very difficult. That is why any substances with hydrophilic moiety is very hard to pass the plasma membrane, and they need some transport support. Okay, only then it is possible. So, students, we will end here, and I will see you in tomorrow's class, and we'll discuss the further topics. Is it okay? That means on Thursday. Students, are you there? I think I'll get a response for this at least. Yeah.